Welcome to the video about classification and binomial nomenclature. While you're watching this video, please take notes on the first section, and then there is a question sheet attached to this file on Blackboard, and you need to answer the questions on that for the speciation section of this video. You will be allowed to use these notes on the next test, so make sure that you take thorough notes. Stop when you need to, stop the video when you need to, and add what you need to do to it. Okay? So let's take a look at this. The first thing that you should do is you should create a mnemonic that will help you remember the Linnaean classification hierarchy. It's called the Linnaean classification hierarchy because it was named after the scientist Linnaeus who came up with this. What this is it's a classification system that we use for all organisms. We start with the domain, and there are three domains: bacteria, archaea, and eukarya. Okay? These are the largest parts of our classification system. Once we are in our domain, we can then say what kingdom the organism is in. There are 6 or more kingdoms depending on how you classify things and depending on what source you're looking at. 4 of the kingdoms are in the eukarya domain. So this here, the eukarya domain, has 4 different kingdoms in it: animals, plants, fungus, and protists. Now, protists is one of the kingdoms that is questionable because most people no longer count protists as one kingdom. Basically, it has been used in the past as a group for organisms that don't fit nicely into animals, plants, or fungus kingdoms. But now that we have some more technology where we can look at these organisms a little bit more detail, they're trying to decide how they should reclassify those organisms. So stay tuned. You might find something. They might pick something soon. So there are 4 kingdoms in the eukarya. There is 1 in bacteria and 1 in archaea. As we get into our kingdoms, we then subclassify our kingdoms to phylum. There are dozens of phylum, and we will study many of them. Within each phylum, there are classes, and there are hundreds of classes. Within each class, there are orders. Within each order, there is a family. Within each family, there are genus. Within each genus, there is species. Now, you notice that as we go down from the most general classification of the domain down to the most specific of the species, the numbers of them increase. Where when we get to the species, there are nearly 2 million described species, and they estimate there's more than 10 million yet to be discovered. So this is a huge amount. So now, by creating a mnemonic is where you use the first letter of each of these words, the ones that I've underlined in blue, and you create a sentence out of it. For example, "Dear King Philip, come over for great spices." Okay, doesn't quite make sense. You can come up with something better, I am sure. So take a minute, write down this classification hierarchy in this order, and come up with a mnemonic so you will remember it. If we take a look at the kingdoms in general, and this is a nice way to look at it, we have these eubacteria here. We have these archaebacteria, so these are the bacteria, different types of bacteria. They are both what is called prokaryotic cells. Prokaryotic cells are cells that do not have their own nucleus. Okay, they have nucleic material inside them, but they don't have their nucleus, which is the control system for the cell. After you have these, we have the protista, and this one does still call the protist their own kingdom. These are what are called eukaryotic cells. Eukaryotic cells mean that they have a bound nucleus. So they have a nucleus, whereas the prokaryotics do not. They think that eukaryotic cells evolved later than prokaryotic cells, and they evolved by one prokaryotic cell trying to eat another one, and it was being unsuccessful. From the Protista, we then have three other kingdoms. We have the plant kingdom, the animal kingdom, and the fungi kingdom. Notice that they are all multicellular. They're all much more complex. They are also all eukaryotic because they all have bound nuclei, which helps them become multicellular organisms. Once we have our organisms fit into the classification system, we have to be able to name our organism specifically enough so that we understand its relationship to other organisms that we are aware of. And that gets us to what's called binomial nomenclature. Binomial means two, nomenclature means naming. So it's a two-word naming system. Each organism that a scientist has discovered has its own scientific name, along with at least one common name. The genus and its species is stated, giving it a specific two-word name. So out of all the parts of the classification system, it is the genus and the species that are most important in naming it, because that's the most specific. This is called the binomial nomenclature. For example, humans are called Homo sapiens. Notice how this is written. This is italicized or it's written with an underline. Also notice that the first letter of the first word is capitalized, but the first letter of the second word is not. Okay? Homo is our genus. Sapiens is our species. So we are Homo sapiens. Earlier humans are also in the same genus, so they start their name with Homo as well. Homo erectus, for example, is an earlier form of a human. So again, notice the formatting of the two words. When you are writing this, you use both words the first time you mention an organism, whether it be in a lab report or in an article review or just answering a question. And then you abbreviate the genus and write the species. So for Homo sapiens, you would write Homo sapiens out the first time, and then after that, just a capital H and a lowercase sapiens, either italicized or underlined. And again, that's how it's done, so you have to remember that. So take a minute in your notes. Again, pause the video here. Take a minute and write the common and scientific names of as many different organisms as you can. You should be able to write down something about a cricket, something about the brassica, and maybe a couple of others that we have talked about over the past few weeks, because we have mentioned scientific names before. You just may not have noticed them. This is the end of the first section of the video, classification and binomial nomenclature. This is the basics of how you need to name things, okay, and how we classify things. Now I want to talk about speciation and what that is and how we deal with that. Speciation, what are species anyways, and how do new ones evolve? Really, that's what we're looking at here. So here we're going to explore different ways to define a species. We're going to learn about various processes through which speciation can occur. And we are going to look at co-speciation, when two lineages split at the same time with one another, and how this can work with plants as well as with animals. So, for example, let's start with defining a species. A species is often defined as a group of individuals that actually or potentially interbreed in nature. That doesn't mean they have to, it just means that they could if they wanted to. So in this sense, a species is the biggest gene pool possible under natural conditions. Keep in mind that from here you should be trying to answer the questions from Blackboard along the way. Okay, you should print that paper out and try and answer these questions. So this is our most generic definition of a species. If a group of individuals can interbreed, whether they do or not is irrelevant. If they can interbreed, they are a species. Okay. For example, These are happy-faced spiders. They look very different, but they can interbreed. So they are the same species. They are the species Theridion, or rather they are Theridion gralator. So their species is the gralator. This definition of species may seem cut and dry. So it may seem like, yeah, of course that makes sense, but it is not. In nature, there are lots of places where it is difficult to apply this definition to a group of organisms because of the way that they reproduce or because of other peculiarities about the organisms. So while these happy-faced spiders are all the same species because they can reproduce, they can mate, that's not always the easiest way to do it. For example, this is a bacterium. Bacterium is singular for bacteria. Oh, that's very weird. Sorry about that. That should say bacterium. This bacterium is shown to be reproducing asexually by binary fission. You can see here and here where the bacterium is starting to pinch its center in, and this will continue, and it will separate the bacterium into two through binary fission. A definition of species as a group of interbreeding individuals cannot be applied to an organism that reproduces only or mainly asexually, because there's no interbreeding going on. It's just the organism becoming the next generation. So that definition that I just gave you of a species doesn't fit here. What about hybrids? Where do they fit in? Many plants, some animals form hybrids in nature. For example, these guys here, the hooded crows and carrion crows, the two on the top, look different. And for the most part, they will typically stay separated. They will typically just mate with their own kind of crow. However, in some areas, they do hybridize. And when they do, their offspring are then neither carrion nor hooded. So you can look at the coloration difference here on the chest of this one, and on this one it has both the chest and the back of the neck feathers looking different. So we have these hybrids. Should they be considered the same species, or should they be considered a different species? And which species should they be considered? So when we look at birds, in this case, for example, we're not sure what to do with them. If we take oak trees, for example, two different types of oak trees are planted next to each other. They often look quite different, but occasionally they are pollinated in such a way that hybrids are formed of each other. How do we count them? Are their offspring a new species? There are lots of other places where the boundary of species is also blurred. So it's not too surprising that we have difficulty with some of this. It's also not too surprising because the idea of species is something that we humans invented for our own convenience. We only did it so we could organize the organisms in the world because we like to think that we control everything. So there's other ways to think about species. One of them is a biological species concept. This is a whole big, broad idea that you need to think about all the different parts for it. One of the things you need to think about with the biological species concept is that appearance isn't everything, okay? So while appearance is helpful in identifying species, it cannot be used to define a species because organisms that may appear to be alike could be from different species. For example, the western meadowlark here on the left and the eastern meadowlark on the right appear to be identical, with the exception of the fact that maybe this guy is a little bit chunkier here, so he's had a little bit more to eat. They appear to be identical. They have the same colorations. One's a little bit bigger. Their ranges overlap, which means that they live in the same areas, but here's a very interesting thing about them. Their distinct songs prevent interbreeding. So when the male sings, when the western meadowlark male sings, the eastern meadowlark female is not interested. So they do not breed, which means then that they cannot be considered to be the same species according to this. Another example, organisms may look very different and yet be the same species. If we look at these ants, you might think that they are distantly related species. In fact, they are sisters. They are two ants of the species Phaidole barbata, and they fulfill different roles in the same colony. So they are the same species, but they don't look anything alike. So keep in mind that many characteristics can vary within a single species. For example, if you've heard of the plant the hydrangea, it can have pink flowers, but the flowers aren't actually flowers, they're actually just leaves, or blue flowers. But that doesn't mean that we can classify the two different forms of different species. In fact, you can cause the blue plants to become pink plants just by changing the type of soil they are in and the amount of aluminum that the plant takes in. So there's a case where they look completely different, but they are exactly the same species. So when we're looking at the biological concept of species, we have to be careful because looks are deceiving. Okay, keep that in mind. And then there are a few other things as well that are problems here. So, adding to the problem, we have already pointed out two of the difficulties with biological species concept. What do you do with asexual organisms? And what do you do with organisms that occasionally form hybrids with one another? But here are some other problems. What is meant by the term at the beginning there that I said that organisms need to be potentially interbreeding? Oops, I forgot an E. What does that mean by potentially interbreeding? Let's take this population of frogs, for example. What if this population of frogs is divided by the highway, as shown here, and it prevents the two groups of frogs from interbreeding? They would interbreed if the highway wasn't there, but because they don't, should we say that they are separate species? Because of this, there's no interbreeding? Is there the potential for interbreeding? Yeah, there is. They could certainly try to cross the highway. They may not make it, but they could try. But we probably shouldn't classify these as different species because they could potentially interbreed even though they don't. But the question then becomes, how distantly separated do they have to be before we make them a new species, before we classify them as a new species? We also have these things called ring species that cause problems. Ring species are species with a geographical distribution that forms a ring and overlaps at the end. So you can see here that there are many subspecies of this sort of salamander. It is from the genus Ensatina. Let me put that down here. They live in California. They exhibit subtle morphological and genetic differences all along their range. In other words, they look a little bit different, and there's a little bit different in their difference. There are a few differences in their DNA, but they all interbreed with their immediate neighbors, with one exception. Where the extreme ends of the range overlap in Southern California, these two guys here do not interbreed. So is that the point where we say, well, that means that they're two different species, even though these guys interbreed, these guys interbreed, these guys interbreed, these guys interbreed, these two interbreed, these two have the possibility of interbreeding right about here, these two interbreed, but these two don't. So does that mean that they're different species? Where do we make that mark for the particular point of speciation? There's another problem as well that we need to talk about. Chronospecies. Chronospecies are different stages in the same evolving lineage that existed at different points in time. So, for example, these trilobites. This trilobite lineage below evolved gradually over time. This obviously presents a problem for the biological species concept. For example, it's not really possible or very meaningful to figure out whether a trilobite that lived maybe here, now let's see, that lived maybe here at 300 million years ago, whether he interbred with its ancestors that lived here at 310 million years ago. That just doesn't even make sense. But because they're a chronospecies, they're evolving and existing at different points in time, should we consider A to be separate from D? Or should we consider them to be the same species? And if we do decide that they're different, where do we split it? Do we say, oh, well, B and A are the same species, but C and D aren't? Or do we say, oh, no, A is definitely by itself, but B and C are the same? There's so many different ways we could look at this that it's really not a clear-cut decision when we're talking about the biological concept, species concept, which is why we have other species concepts as well, because of the limitations of the biological species concept. And it does work, and it has been very influential in the growth of the evolutionary theory, but we have to come up with other species concepts to clarify some of these limitations. So we also have something called a recognition species concept. What this means is that a species is a set of organisms that can recognize each other as potential mates. So it's sort of like the potential interbreeding, but it doesn't say they have to, it just has to say that they recognize each other. So even though these two frogs have been prevented from mating, the fact that they recognize each other as a potential mate makes them the same species under the recognition species concept. I don't know how you would test this. The phonetic species concept is a species is a set of organisms that are phenotypically similar and look different from other sets of organisms. Phenotypically means that they look the same. Your phenotype is what you look like. So your physical looks. According to this concept, phenotypic similarity is all that matters in recognizing separate species. Since the frogs depicted here look the same, even though they're prevented from mating with each other, they would be considered the same species according to phonetic species concept. Now again, you could see where that could be an issue like previously when we were talking about the meadowlarks. We also have something called phylogenetic species concept. A species is at a tip of a phylogeny. This picture here, this is called a phylogeny. And basically that is a fancy name to say that you have a family tree. Okay, so a species in this case is only at the tip. That is, it's the smallest set of organisms that share an ancestor and can be distinguished from other such sets. Under this definition, a ring species is a single species that encompasses a lot of phenotypic variation. So that ring species that we saw before, they're all one species. And this guy is a separate species, and this guy is a separate species. So if we look at this example here, the salamander in lineage A and the salamander in lineage B are different species, but they each have a common ancestor that individuals of other species do not. Even though it has diversified a lot, lineage C is a single species according to this. And none of the subspecies in lineage C have a single common ancestor separate from other subspecies. In other words, they all follow back the family tree. They all come back to this point right here. And that is the same common ancestor as the organism in B. And then this common ancestor, if we go back in time, had the same common ancestor as A did. Okay, so what this means then is that the organisms in B and C are more closely related than the organisms in A and B because they have a more recent common ancestor. I forgot the end. Okay. A researcher's choice of species concepts often reflects his or her research focus. Making that decision requires the scientist to commit to a species concept. For most purposes and for communication with the general public, the biological species concept is used. However, there are times that scientists don't use that. So let's talk more about what speciation actually is and how do you even get these different species. So we have to define speciation. Speciation is a lineage splitting event. That produces two or more separate species. Imagine that you're looking at the tip of the tree of life that constitutes a species of fruit flies, like what's shown. You move down the phylogeny to where your fruit fly twig is connected to the rest of the tree, like here. That branching point and every other branching point on the tree is a speciation event. At that point, genetic changes resulted in two separate fruit fly lineages, which previously had just been one lineage. But why and how did this happen? So what occurred here? What occurred here that caused these genetic differences? At this point, we really don't know because some of these occurred quite a long time ago. But let's look at an example of how this could have happened, and I'll give you a general understanding of how this could have happened with a fruit fly population. So let's set the scene. We have a population of wild fruit flies minding its own business on several bunches of rotting bananas. They are cheerfully laying their eggs in the mushy fruit. But then disaster strikes. A hurricane washes the bananas and the immature fruit flies they contain out to sea. So it has come across the sea. The banana bunch eventually washes up on an island off the coast of the mainland. This is where the mommy and daddy were. The fruit flies mature and emerge from their slimy nursery onto the lonely island. At this point in time, the two portions of the population, the one on the mainland over here and the one on the island over here, are too far apart for gene flow to unite them, so they could not mate. But at this point, speciation has not yet occurred. Any fruit flies that go back to the mainland could mate and produce healthy offspring with the mainland flies. Right now, they are just separated, but they haven't made any changes yet. So no speciation has occurred. Ecological conditions are slightly different on the island, and the island population evolves under different selective pressures and experiences different random events than the mainland population does. Morphology, meaning changes, food preferences, and courtship displays change over the course of many generations of natural selection. So you can now see that over here we have our island group, and here we have our mainland. And you can notice some changes occurring already on them because there's been many generations of natural selection going on. So the populations have diverged. When another storm reintroduces the island flies to the mainland, they will not readily mate with the mainland flies since they've evolved different courtship behaviors. So these guys aren't going to get together because they don't understand what's going on. The few that do mate with mainland flies produce inviable eggs, meaning eggs that cannot reproduce or cannot survive, because of other genetic differences between the two populations. The lineage has split now that genes cannot flow between the two populations. So this is a simplified model of speciation by geographic isolation. But it gives an idea of some of the processes that might work in speciation. In most real-life cases, however, we can only put together part of the story from the available evidence. However, the evidence is typically pretty strong to make the case for what actually happened. So just let's recap here. The causes of speciation. Number one cause is geographic isolation, like the fruit flies. The scientists think the geographic isolation is the common way for things to happen. Or the common cause, if you want to say that. A river may change its course, or a mountain may rise up, continents drift, organisms migrate, whatever. At some point in time, a continuous population was divided into two or more smaller populations. Even if there wasn't a physical barrier between them, something separated them. So that over time, their natural selection causes them to become two separate organisms, and they can no longer mate because their genetics are different. That's one way. That's the most common way. Speciation might also occur in a population with no specific reason, it seems. Imagine a situation in which a population extends over a broad geographic range. So in other words, they live in a wide area. Mating throughout the population is not random. Individuals in the far west would have a 0 chance of mating with individuals in the far eastern edge of the range. So we have reduced the gene flow, or how the genetics move back and forth, not by total isolation, but simply because they weren't around. So there may or may not be sufficient change or reduction of gene flow to cause speciation. It would probably alter the gene frequency in the group at different ends of the range, so much that they would not be able to mate if they were reunited. But that would take quite a long time. So that's what this one shows, the reduction of gene flow. This is often due to very large areas that the population lives on. So, if you're this guy right here, then chances are you're going to mate with somebody that's in this general vicinity, right? Very rarely are you going to even get to see this person over here or this bug over here. Forget about have a chance to mate. So even though there's no physical barrier between there, you can see here that the chances of mating with somebody way over here is close to none. So the result would be you would have these hybrids. And the population over here may eventually turn purple, the population over here may eventually turn green. The population in the middle would be sort of the hybrid between them. Eventually, you could end up with a case of that splitting into two species because of the reduction of gene flow. So the most common cause of speciation is genetic isolation due to geographic isolation and the reduction of gene flow. Hopefully, this gives you a better understanding of how species come about. Make sure you try to answer all of those questions, and we will talk more about this in class tomorrow. Bye for now.